Good morning, everyone, and welcome to today's presentation. I'm Dr. S.K. Singh, and I'm excited to guide you through this session. Today, we'll be focusing on the calibration of the FTIR spectrophotometer. Over the next few slides, we'll cover the key steps involved in calibration, why it's important, and how to ensure accurate measurements. Let's get started and explore this essential aspect of spectrophotometry together. Welcome to our second slide, where we dive into the basics of Fourier transform infrared, or FTIR, spectroscopy. This technique is absolutely essential in our field because it allows us to analyze materials with high precision. It's primarily used for identifying raw materials, making sure that the ingredients we use are authentic and of the right quality. It also helps verify the identity of active pharmaceutical ingredients, or APIs, and excipients, ensuring everything matches our specifications. Additionally, FTIR is invaluable for functional group analysis, which means we can determine the specific chemical groups present in a sample, crucial for understanding molecular structure. And of course, it plays a key role in GMP compliance testing, helping us meet regulatory standards and maintain product quality. But here, the key point, accurate calibration of our FTIR instruments is vital. Proper calibration guarantees that the spectral data we gather is reliable and consistent, which is essential for regulatory compliance and for making sound scientific decisions. So, in essence, FTIR spectroscopy underpins many critical aspects of quality control and assurance in our processes, ensuring we deliver safe, effective, and compliant products. Let's take a closer look at the applicable guidelines that govern FTIR calibration. First, USP 854 focuses on mid-infrared spectroscopy, outlining standards for accuracy and reliability in spectral measurements. Then, we have USP 1058, which deals with the qualification of analytical instruments, ensuring they perform consistently over time. ICHQ2, R1, is another critical guideline that emphasizes the concepts of accuracy and precision, fundamental to obtaining valid results. Moving to regulatory requirements, 21 CFR Part 211 specifies the importance of instrument calibration and control to maintain product quality. Similarly, EU GMP Annex 15 provides guidance on qualification and calibration processes, aligning with these standards globally. Now, why is calibration so important for FTIR? Its main purpose is to verify that the wavenumber readings are accurate, which is vital for correctly identifying and quantifying substances. It also confirms the instrument's resolution performance, ensuring it can distinguish between closely spaced signals. Calibration helps us maintain photometric, or energy, accuracy so that absorbance measurements are reliable. Additionally, it allows us to detect any instrument drift over time, which could otherwise lead to inconsistent data. Lastly, proper calibration plays a key role in maintaining data integrity, aligning with the ALCOA Plus principles: attributable, legible, contemporaneous, original, accurate, plus the additional qualities of complete, consistent, enduring, and available. All these points connect to our broader goal of ensuring accurate, reliable, and compliant analytical results. Let's take a closer look at the calibration parameters outlined by USP 854. These are critical for ensuring our spectroscopic measurements are accurate and reliable. First, we have wavenumber accuracy, which essentially tells us how precise our spectral measurements are in terms of the wavenumber scale. It's important because even small deviations can impact our identification of compounds. Next, resolution is key. This defines how well our instrument can distinguish between two closely spaced absorption bands. Higher resolution means more detailed spectral data. Then, there's photometric accuracy, or what we call the energy ratio. This measures how accurately our instrument records the intensity of the spectral signals, which is crucial for quantitative analysis. We also need to consider the signal-to-noise ratio, or SN. This tells us how clearly our signal stands out against the background noise, impacting the sensitivity of our measurements. Lastly, baseline stability, when applicable, ensures that our spectral baseline remains consistent over time, which is vital for detecting subtle spectral features without drift or fluctuation. All these parameters together form the foundation of a well-calibrated, trustworthy spectroscopic method, aligning perfectly with our goal of ensuring high-quality, reproducible results in our analyses. All right, let's talk about wave number accuracy. The main objective here is to verify that the infrared absorption peak positions are correctly assigned. In other words, we want to make sure that the peaks we're seeing actually correspond to the right vibrations in the sample. To do this, we use a standard, which in this case is a polystyrene film, and it's an USB reference standard. This standard has certified peaks at specific wave numbers, namely 1601, 1583, 1154, 1028, 906, and 841 centimeters inverse. These known peaks serve as our reference points. By comparing our measured peak positions to these certified standards, we can confirm whether our instrument is accurately capturing the absorption peaks. This step is crucial because accurate peak assignment ensures the reliability of our spectral analysis and subsequent interpretations. So, in the context of our overall goal to ensure quality and precision, verifying wave number accuracy helps us maintain confidence in our spectroscopic measurements and the data we generate. All right, let's walk through the procedure for checking wave number accuracy. First, we place a polystyrene film directly in the IR beam path. This film is our standard for calibration because it has well-defined absorption peaks. Next, we scan the spectrum over the range from 4,000 to 400 centimeters to the -1. Once we have the spectrum, we identify the observed peak positions. These are the points where the absorption peaks appear in our data. Then, we compare these observed peak positions with the certified, or known, values for polystyrene. This comparison helps us see how accurate our instrument is. After that, we calculate the deviation for each peak, which tells us how far off our measurements are from the standard. These steps are crucial because they ensure our spectrometer provides precise and reliable data, which is fundamental for accurate analysis throughout the entire process. Let's take a closer look at wave number accuracy, starting with the formula. Essentially, this helps us quantify how precise our measurements are. The key idea is that the accuracy depends on how well we can determine the position of a spectral line relative to its true value. When we talk about the formula, it typically involves the difference between the measured wave number and the actual or reference wave number, divided by the reference, often expressed in parts per million or similar units. This gives us a clear, numerical way to assess our measurements' accuracy. Now, to make this more concrete, let's walk through an example. Suppose we measure a spectral line at a wave number of 2000.005 cm1, but the known reference value is 2000 cm1. The difference is 0.005 cm1. Applying the formula, we divide that difference by the reference value, which gives us 0.00000025. Multiplying by 1,000,000 converts this into parts per million, resulting in 2.5 ppm. This example illustrates how small deviations can be quantified precisely, and it highlights the importance of understanding and calculating this accuracy for reliable spectral analysis. Connecting this back to the overall theme, mastering wave number accuracy ensures our measurements are trustworthy, which is crucial for applications ranging from material characterization to environmental monitoring. Now, let's move on to our resolution test. The main objective here is to verify how well the FTIR can separate peaks that are very close together. This is a critical aspect because in many real-world samples, peaks often appear very close, and the instrument's ability to distinguish them is essential for accurate analysis. For our standard, we'll be using a polystyrene film. It's a common reference material because its spectral features are well known and consistent, making it ideal for testing resolution. The procedure involves measuring the absorbance at two specific points. First at 2870 centimeters inverse, which corresponds to the peak, and then at 2849 centimeters inverse, which is the valley between the peaks. By analyzing these measurements, we can determine if our FTIR instrument can clearly resolve these two features. Remember, the goal is to see if the instrument can distinguish between peaks that are very close in frequency, ensuring our analysis will be precise even with complex samples. Let's take a closer look at the concept of resolution, specifically focusing on formula and acceptance. When we talk about resolution in this context, we're referring to how we can find clarity and solutions through structured approaches, like formulas, that help us process complex situations. These formulas serve as tools to simplify and understand the core issues, making it easier to develop effective responses. Acceptance, on the other hand, is about acknowledging reality as it is, without resistance or denial. It's a crucial part of resolution because recognizing what we cannot change allows us to redirect our energy toward constructive actions. Combining these two, using formulas to analyze and acceptance to embrace the situation, creates a powerful framework for moving forward. This approach ties into our overall theme of growth and resilience. By applying clear formulas and cultivating acceptance, we can navigate challenges more effectively, turning obstacles into opportunities for learning and progress. It's about balancing understanding with peace and action with patience. Let's talk about photometric accuracy, specifically focusing on the energy ratio. When we refer to photometric accuracy, we're essentially measuring how precisely our system can estimate the amount of light energy coming from a scene. The energy ratio is a key metric here. It compares the energy captured or measured by our system to the actual energy present in the scene. A high energy ratio indicates that our system is very accurate, capturing most of the light energy without significant loss or distortion. Conversely, a lower ratio suggests there might be some inaccuracies or inefficiencies in measurement. Understanding and optimizing this ratio is crucial because it directly impacts the reliability of our data, especially in applications like remote sensing or imaging, where precise light measurements are essential. Throughout this presentation, we've emphasized the importance of accurate data collection. Improving photometric accuracy ensures that our analysis and conclusions are based on trustworthy information, ultimately strengthening the overall quality of our work. Let's talk about the signal-to-noise ratio, or SN for short. This is a really important concept because it helps us understand how well a signal can be distinguished from background noise. Essentially, a higher SN ratio means that the signal stands out clearly against the noise, making it easier to analyze or interpret. Conversely, if the SN ratio is low, the noise can obscure the signal, which can lead to errors or misinterpretations. In many fields, whether we're dealing with communications, data analysis, or even imaging, optimizing the SN ratio is crucial for accuracy and reliability. Throughout this presentation, we've been emphasizing the importance of clear, accurate data, and understanding SN helps us quantify just how clear or noisy our signals are. So, when designing systems or conducting experiments, always keep an eye on the SN ratio. It's a key indicator of the quality of your data. Now, let's talk about calibration frequency and documentation, which are crucial for maintaining reliable analytical results. First, regarding frequency, calibration should be performed at the initial qualification to establish a baseline. Then, regular checks are necessary, typically every 6 or 12 months, to ensure continued accuracy. Additionally, calibration should be repeated after any major maintenance or if the equipment has been relocated, since these events can affect instrument performance. Moving on to documentation, it's essential to keep a thorough record of every calibration. This includes the calibration protocol, which outlines the steps taken, and the raw spectra obtained during calibration. You also want to record the calculations and results, so there's a clear trail showing how the calibration was verified. Importantly, each calibration should include a pass or fail statement, indicating whether the instrument is within acceptable limits. And finally, signatures from the analyst and reviewer are vital for accountability and validation. Overall, proper calibration frequency and meticulous documentation help ensure the integrity and reproducibility of your data. which is central to maintaining confidence in your analytical processes. Now, let's talk about how we handle out-of-tolerance, or OOT, situations. When a measurement or instrument falls outside the acceptable range, the first step is to clearly mark it as "Do Not Use." This helps prevent any accidental use of potentially inaccurate data. Next, we conduct a root cause investigation to understand why the instrument was out of tolerance, whether it's a calibration issue, environmental factors, or equipment malfunction. Once we identify the cause, we assess the impact on the tested samples to determine if any results need to be retested or if there might be implications for our overall quality. Based on that, we take corrective actions, which often include recalibrating the instrument or fixing the underlying issue. After that, we perform a recalibration to ensure everything is back within specifications. Before we can reuse the instrument, our QA team reviews and approves the corrective actions and recalibration. This process is crucial to maintaining the integrity and accuracy of our testing. It's all about making sure that even when something goes wrong, we have a structured way to address it quickly and effectively, supporting our overall commitment to quality and reliability. Let's wrap up with some key takeaways and best practices to ensure we maintain high standards. First, always use USB reference standards. These are critical for ensuring the accuracy and consistency of your measurements. Next, follow validated SOPs, standard operating procedures that have been tested and proven effective, to keep your processes reliable and compliant. It's also essential to maintain traceability and be audit-ready at all times. This means documenting everything thoroughly so that your data can be verified and audited easily. Lastly, calibration plays a vital role in supporting data integrity and GMP compliance. Regular calibration ensures your instruments are accurate, which directly impacts the quality and reliability of your results. By integrating these practices, we reinforce the overall quality system and uphold the integrity of our work. For further support, please feel free to contact us. We're here to help with any questions or additional information you might need after this presentation. Thank you so much for your time and attention today. Your engagement is truly appreciated, and I hope you found this session valuable. Remember, we're always available to assist you as you apply these concepts or explore next steps. Again, thank you, and have a great day.